Hello, and welcome to this installment of the Future Grind podcast. I'm your host, Ryan O'Shea, and today I'll be speaking with Aaron Traywick, the CEO of Ascendance Biomedical. Aaron Traywick is a controversial figure that seeks to democratize access to unregulated, experimental, and some would say potentially dangerous medical treatments and enhancements. Aaron recently made headlines when he injected himself with an experimental herpes treatment while on stage at the BDYHAX Human Augmentation Conference in Austin. He hopes to prove these treatments safe and then make them available to others who would like to access them. I will remind everyone that self-administering any type of unregulated gene therapy or other treatment is risky. Anything that Aaron mentions during this interview should be viewed as an experimental research compound not intended for human use. I encourage you to consult your doctor with any questions. Despite the controversy surrounding Aaron and his work, I feel that this conversation is an important one to have. I was very interested in his background and the philosophy behind his work, and I think that you will be as well. For better or worse, DIY gene therapy will only continue to grow. Aaron has positioned himself near the forefront of this movement, and I think we'd all be better served if we understood more about it. As always, show notes and more are available at futuregrind.org. We're on iTunes, Stitcher, and wherever podcasts are found. A video version of this episode is available on Facebook and YouTube. Make sure to subscribe. You can also like, comment, and share to spread the word. This is Future Grind. All right, and now we are here with Aaron Traywick. Aaron, thanks for joining us.
Hey, thanks for having me, Ryan.
So what first got you interested in tackling the problem of aging?
Well, as a child, as a teenager, I worked to take care of my grandparents. When we were growing up there in Alabama, they were going through a lot of health issues related to diabetes and really facing a long and slow death from diabetes-related issues. What it came down to was, one day my grandma was in the hospital and she was in the ventilator, and the doctors come in and they tell you there's nothing they can do for her. And if you give them this much money every day, they can keep her alive as long as you want. And she may recover even, and she may not. That kind of dichotomy, that kind of paradigm to me was something I didn't want to ever have to deal with or live with again. I didn't ever want to be in the position that my parents were in for their parents. I didn't ever want to have to be in that position to make those kinds of decisions for them. And I was disgusted at the idea that if I ever was in that position, then I would have to be making financial decisions based on my own self-preservation at the cost of my loved one's life. And then to also have to think about things like inheritance and property that would be transferred in the event of their death. And all of these things to me were just disgusting. Like disgusting base elements of life in this species that we should rise above. There are certain behaviors in this species that we deem criminal. We have general respect for each other's property and each other's bodies, and punishments for when those things are transgressed upon. And yet we allow this behavior. Like we allow ourselves to feel as though this is not an act of violence towards those who truly love us the most. They are the ones who literally are the only reason we are here. So that is mainly why.
And how old were you when this was happening, when you went through these experiences?
I was 16.
So still pretty young. And how did you find out that there were communities of people already working to tackle death and the diseases of aging? What was your first introduction to that world?
I spent some time after doing a lot of community organizing work and establishing a decent career for myself in that area around public health and the environment in Alabama. I traveled very frequently through South America, studying the works of Robert Anton Wilson and Timothy Leary, trying to understand a little bit more about, Timothy Leary's descriptions of where the psychological development and evolution of humanity was headed. Space migration, intelligence expansion, life extension. And the life extension one was the one that appealed to me the most. It seemed to me Like, this is the one that was the real barrier for all of the other ones. And that if this one was surmounted in some way, not only would the individual pouring their work into that area have the opportunity to impact the greatest number of human lives and suffering as possible as compared to any other sort of work, they would also be creating a world where they could feel safer and not so shucked and cajoled and tossed around by the pressures that come with living an existence that is perpetually required to deny the inevitability of its own demise in order to function.
At this point, I've spoken to a number of individuals who are working to overcome biological limitations, and I always ask what first got them started on that path. And the most frequent answers I get are an experience with illness or death in childhood, or experiences had while traveling. And it seems that you've had both, so that's interesting. I recently found out that both you and I worked on Capitol Hill at the same time in the fall of 2013. While you were serving in the office of a congresswoman from Colorado, I was in the office of a representative from the Pittsburgh area. But while I returned to Pittsburgh to work with Grindhouse Wetware, you stuck around. And in 2015, in Washington, D.C., you co-founded the Global Healthspan Policy Institute, which is a nonprofit think tank with the goal of advancing the development of interventions for aging-related disease. How did that get its start, and what were you able to accomplish with that?
So much of that work owes its great success to not just myself, but to the collaborators that were at the core of it. Edwina Rogers, a woman with over 25 years of great experience as a professional lobbyist and as a public policy staff person in multiple presidential and congressional administrations. And Kevin Parrott, a very well-connected co-founder of the Methuselah Foundation and sends research to organizations at the forefront of all life extension research these days. And through our connections, we decided that we wanted to form the Institute. We wanted to make it a reality, and we set about finding the chief luminaries in the field of aging research and its underlying causes and conditions. Nir Barzilai, Judith Campisi, Gordon Lithgow, J.L. Shansky. And we had them all come, and the fellows of our think tank, and we represented them there on Capitol Hill. And what we specifically did was we worked to secure $26 million for the first FDA greenlit trials to treat aging as a disease through the use of metformin, which is the world's preferred drug of choice for the treatment of diabetes, not so much in the southern states or the Midwest, where insulin is heavily advertised, though it's much less effective and sometimes even potentially deadly. But of course, metformin is a generic. It is not really owned or advertised for by any major pharmaceutical company. So its effects in reducing multiple cancer risks for multiple cancers, cardiovascular disease and health, and mitigating other interesting elements of the aging process have been frequently overlooked. And of course, Edwina and I, we're family, we're cousins. And so for us, the diabetes issue was a primary passion point. It made sense to us. We'd been through the experience with our grandparents, and we knew that we didn't want that for others. And while ultimately the acquisition that we worked on with Senate Minority Leader Schumer and Senator Thad Cochran of the Appropriations Committee in a very bipartisan project did not go through and has not yet gone through, we were able to catalyze enough enthusiasm publicly to generate $9 million of private investment that have been able to launch the pilot trials for the clinical trials themselves, which we're very proud of, and we're very glad that the ball is moving forward. Metformin is a really low-hanging fruit compared to a lot of the other work that we're doing in gene therapy. It is far from, the most ideal and breakthrough treatment, but it is one of the most mainstream, and it's something that is very accessible to most any person in the world. It's a deep structural issue that the Institute was founded to address. And that is that, in the early 70s when Ted Kennedy was, going around and raising funding for all these different diseases, what he would do is he would come in with a think tank, and he would parade them in front of all the congresspersons and say, here's Alzheimer's disease. Here's all the ways that this is affecting everyday Americans, and here's why we need to found an institute in the National Institute of Health specifically designed to target and eliminate this disease. And he did that as a model over and over and over again for cancer, for a variety of other diseases. And that was a very effective model at its time. Now, we're in a place where we have all these different institutes of health all competing for federal funding and all competing for nonprofit and public engagement around finding a cure for a specific disease. And we know very clearly that there are underlying mechanisms that result in all of these diseases, that if we target them, we can have a positive effect on all the diseases at once. However, there's no institute for that. There's the National Institute of Aging, but something like less than 1% of 1%, or some very, very, very small number of all federal health-related dollars that actually goes to research designed specifically to create therapeutic interventions in the aging process, in the underlying causes of aging-related disease. That research has promoted and provided some very outstanding work. However, it's ridiculous that it is given such little support. And so it was necessary to speak to the members of Congress and to have them understand the reason why we're having such trouble funding this is because the government is not set up to have a place for funding this. And that's ultimately why we worked with the military, because the military does provide opportunities for fast tracking and expediting innovative research. So it's a deep structural issue in terms of the way that our federal funding dollars are distributed towards these academies. And obviously, you can't speak or have the researchers in those divisions who are studying and tackling those diseases speak to those issues or speak to the value of targeting something that would effectively intervene in all the diseases or at least more than one, because they are tied to federal funding, to money that pays their bills and pays for their livelihoods and pays for their research. And they're not going to rock the boat. That's just not human nature. And it's certainly not the nature of a department head or a division head of any of these agencies or anyone involved in the bureaucratic world there in Washington. If money is coming in and it is supporting people's work and livelihood and jobs, then it is deemed that is an appropriate channel.
Are you still involved with the GHPI, or what are they up to now?
Right now, Edwina is handling that. A lot of our team branched off to form this company, Ascendance Biomedical, so that we could tackle breakthrough treatments that we felt we could bring to market and to the individual who wanted them in a much more expeditious way than the standard FDA clinical trial processes, based on our work with the FDA and our understanding of what companies can do and what companies are able to do to expedite treatments for clinical delivery and non-clinical delivery to at-need patient communities.
And you currently serve as the CEO of Ascendance Biomedical, which I imagine is what the majority of our conversation today will focus on. So let's give a little bit of background information about what that company is and what you hope to accomplish with it.
Yeah, so Ascendance Biomedical essentially functions as an accelerator incubator for gene therapy development, targeting infectious disease, terminal and chronic disease, and human augmentation. We have 3 novel open access platforms that provide the opportunity for individual researchers and investors, companies, and academies to engage with our platforms and develop novel applications that can be delivered at an end cost to the patient of less than $100 for a disease-specific contingency and less than $1,000 for an augmentative specific contingency. So as soon as we have the raw human safety and efficacy data for a given therapy, we work to spin that company off into a publicly shared entity as quickly as possible and engage the FDA in the most effective way.
For better or worse, there are a lot of expectations of the CEO of a biomedical company. While anyone can theoretically serve in that role, most people expect certain amounts of prior experience, qualifications, or a specific educational background. What is your background in those areas, and how did it lead you to this career path?
I am from a firmly rooted middle-class family in Alabama. I was adopted at 7 days old. My parents raised me in a very, very small town, one of the smallest in Alabama, right where they filmed Big Fish, actually, if you've ever seen that movie. And my father was a welder in the National Guard, and my mama was a nurse at a rehab clinic, and later worked for the state, still does, for rehabilitation. I went to a very enjoyable and very well-known public liberal arts university in Alabama called the University of Montevallo. And while I was there, I studied whatever I felt like studying. If I wanted to do an internship in business, I just signed up for the class and did that. If I wanted to study philosophy, if I wanted to study sociology, I did that. If I wanted to learn sign language, I did that. And at the end of the day, my passion for community organizing and for engaging local politics, running election campaigns, and running campaigns with other university groups across the state, ultimately I just followed my passions in terms of getting results and helping people and doing my damnedest just to save human lives in whatever way that could be done. And in the process, I gained an education out of it and wrote a thesis for my degree and moved on from there into the world to continue what is essentially and always will be self-directed research.
With your background in politics and policy, and your interest in using science and technology to overcome human limitations, what are your thoughts on the advent of transhumanist political parties?
Well, I've said this many times, and I'll always say it. Transhumanism has the potential to show the world that innovations in technology can alleviate the frustrations, anxieties, and insecurities that have plagued humanity for millennia. Diseases are one of those. Economics are another one of those. The problem of unnecessary labor and work for work's sake, instead of individuals being able to free to pursue the passions and talents that they themselves should always have the opportunity to discover. Transhumanism has the opportunity to, if effectively messaged in a way that can reach the average everyday American and show them that their lives can be made easier, that their lives can be made particularly less economically strenuous, and that just general quality of life can be improved, then transhumanism will win every time, because transhumanism is rooted deeply in, libertarian ideals of strong, principled core values of freedom and the right to pursue self-directed evolution, provided that that does not harm others.
Do you self-identify as a transhumanist or a biohacker?
No. I would consider myself a biohacker only in the sense that Jonas Salk was a biohacker when he self-tested the polio vaccine on himself and his family, saving millions of lives in the process and creating open source information that was then able to spread freely around the world and eradicate the disease in a time frame that could never have occurred had he not made all of those decisions on behalf of himself and humanity. And I consider myself a transhumanist only in the sense that I consider myself a humanist, and that I want only for the strategies and ideologies and creative potentials of any belief system to perpetuate, provided that they give individuals the means by which to live a healthier, happier, longer, and more enjoyable, more blissful life for themselves and their families and all those they love.
You mentioned Jonas Salk there, and I am an alumnus of the University of Pittsburgh, which of course is where Salk pioneered his polio vaccine in the 1950s. And you're right, he did test this potentially dangerous vaccine not only on himself, but also on his children. The story goes that he boiled syringes in his family kitchen before administering the experimental treatment. I imagine many holier-than-thou online commenters would have a lot to say about these actions if Salk were doing this work today, but it worked. The polio vaccine was effective, and countless lives were saved. And Salk didn't patent this vaccine. It was given freely to the world. And I wonder if this would even be possible today. I've worked to bring technology that has been developed with the support of the University of Pittsburgh and other stakeholders to the world, and it's so unreasonably difficult. There is such a desire for financial incentive and a control of intellectual property that I truly feel is hurting humanity overall. The research is great, but if no one can use the finished product, what good does it do? And I say this as someone who loves Pitt and the people there, but something has to change. And from what I've heard at least, it seems that Ascendance Biomedical wants to disrupt the traditional medical and pharmaceutical industries and forever change how treatments are brought to market and the financial incentives involved. What are the problems that you see with the traditional medical system, and how do you hope to solve them?
Well, the problems you've illustrated there very clearly, and they're the same problems that we ourselves will desire to overcome, particularly with our HIV therapy. We are working with a compound that we believe should be made available as emergency use for individuals in parts of the world, such as Venezuela specifically, where no antiretrovirals and no HIV or AIDS medications are being allowed in the country due to the politics of a particular dictatorial government and the relative sloth of the international community to intervene in what is essentially public health genocide for individuals with HIV and AIDS in that country. In the short term, we're working as quickly as possible to ensure that any cures for any diseases that we come up with are available to any member of the public, as a research compound for less than $100 a dose, for what should be a one-time only dose, a one-time only cure, or a one-time only vaccine. And in the very near future, by this time next year, we will have technology that you will be able to have in your home with your computer that you can run as software. You can then take our protocols for how to create a particular gene therapy for a particular disease state. And with the correct quality controlled reagents and chemicals, you will be able to produce that yourself for yourself. So we are dedicated to the idea of making our own production facilities for the promulgation of these therapies obsolete, and in a sense bring to life the idea of the maker, the maker machine that can chemically create anything. It's a great idea. We can figure out all the other stuff later. Let's figure out the medicine thing first, the health thing first, and give every person the means by which to produce everything that they would ever need for their own health.
While those are some ambitious goals and a surprisingly short time frame, I'm sure a lot of people hearing this are a bit skeptical, myself included, but I definitely think these are big problems that we need to solve, and I hope that they are solved. We need to make sure that we do this safely. We want to move quickly and efficiently, but I don't think we want to risk rushing things, which could actually set us back. But I agree, people who want to use experimental treatments and who understand the risks should be able to, especially when the only alternative is death. And you mentioned that your goal is to make some of your research compounds available to people in Venezuela. When I was a student at the University of Pittsburgh, I worked on a Smithsonian Channel documentary about the Salk polio vaccine. It was called A Shot to Save the World, and in that documentary, we focused quite a bit on the fact that Jonas Salk had a lot of fear that his experimental polio vaccine would cause harm to the Pittsburgh area children that it was tested in. And your developments at Ascendance Biomedical are already being tested in humans other than yourself. Tristan Roberts, for example, has self-administered an experimental HIV treatment developed by Ascendance. Do you share Salk's fears of causing harm?
Well, of course. And as you know, I'm having this conversation with you now, just a little over a week of having injected myself with the experimental gene therapy of our own design and creation for the curing of herpes. And it's been a very intense week. Interpreting essentially every aspect of my day-to-day life has been recognized and experienced through a new veil. Everything from the way that I eat to the way that my body appears to react to particular stresses, to just everything generally is just slightly different, and it's noticeably different. It's not something you can forget. It's something is there. Something is changing, you don't know exactly what it is, and you want to take care in how you move forward, because ultimately no one but you can tell what's going on. There is no research. When people are experimenting with these gene therapies, when they're working with them, there is no resource to go to. I mean, the people who tell you to fear self-experimentation, well, of course they're going to say that because they can't say anything else. They can't tell you. They can't predict for you. No one can. There is no real way to truly and novelly interpret this experience except through the function of one's own personal physiological and neurological laboratory, our bodies, our space.
Yeah, let's get into your injection of Ascendance's experimental herpes treatment. This was actually done on stage during your talk at the BDYHAX Human Augmentation Conference in Austin. And I was there during your talk and live streamed the injection on the Future Grind Facebook page. So if anyone hasn't seen it, you can watch what happened by viewing the video there. And your injection received a lot of coverage from media outlets such as Gizmodo, The Verge, MIT Technology Review, BuzzFeed, Men's Health, the BBC, and many more. And this all happened just 1 week ago. You injected yourself in the first week of February, and were here talking during the second week. And it actually ended up being quite controversial. There was some confusion about what it was that you actually injected and how it was supposed to work, whether it was being described as a vaccine or a cure for herpes, and whether it had been adequately tested before you did the injection on stage. So let's clear some of that up. What is it that was injected, and what was it supposed to do?
So what I'm going to do here is I'm going to read directly from our public messaging document for it, because that is the easiest way for us to completely articulate, everything about the experimental vaccine, experimental cure itself. So this is a comprehensive description. So even though research on a herpes vaccine has been going on for almost 30 years, because of the way the virus works, traditional ways of making a vaccine have thus far been ineffective. And our researchers have found previous research that demonstrates the use of a live but modified virus, if administered as a functional vaccine for HSV, has a very good likelihood of being that functional vaccine, that functional cure. The way this works is that on the outside of the virus is a protein called glycoprotein D that both allows it to enter new cells and hide from the immune system. The live virus used in the vaccine has this protein knocked out, so new virus particles can be produced, but the new particles can't enter new cells. Through the use of a transfection agent, some of these defective viruses can be made to enter a patient's cells. These will start to multiply, but because they can't infect new cells or hide from the immune system, the patient's immune cells should be able to quickly find these particles and learn how to kill them. Once they've done that, regular herpes particles in the patient's system should be found by the newly armed immune system, and the virus can be eliminated from the body once and for all. The demonstration at Biohacking Con used an even simpler approach as an initial inquiry to see if it could be an even more effective, viable option. Instead of using live virus particles to deliver the initial DNA that allows defective particles to be produced in the patient, we use the DNA itself and a common transfection agent. If it works, the results should be the same as the live virus test that will be conducted later in individuals who do not have herpes. If it works, live defective virus particles will be produced, immune cells will find them, and then functional virus particles will be eliminated from the body. If it doesn't work, then we'll just proceed forward with the other one for testing. If it does work, however, this will be a novel therapeutic approach that is easier and even cheaper to produce and make available. Well, to the quality control circumstances, obviously, high power electron microscopy and PCR to ensure concentration and quality control standards and things of that nature.
So how have you been doing in the week since the injection, and what are the next steps in this process? When do you think you'll have results to report?
The experience of herpes itself is one where the body is constantly and effectively always at war. Antibodies are always being produced by the body once herpes is in the body to fight the herpes. So that is always ongoing. What we anticipated and what has been my physiological experience appears to be an uptake in the amount of antibodies in my body. By what concentration, we do not know. That uptick may be all that it takes to effectively reduce the overall viral loads within my body. We will know a little bit more on Saturday, since on Wednesday I'll be doing the blood testing that will show the concentration level of the herpes antibodies that my body is producing to fight against herpes. However, that will not be a confirmation of anything except that the vaccine is creating a higher concentration of antibodies in my body to fight. So that will let us know that it is working, but not that it works. To know that it works, for an individual like myself, I don't have any symptoms of herpes. None of my partners do and never have. In fact, partners I have currently don't even have herpes. And that's interesting enough as it is. But in any case, the only effective way for someone who is 100% asymptomatic to test for the presence of herpes within their body is to do daily or every other day swabbing in multiple locations of the body. It doesn't really matter what they are. It can be any location of the body so long as there's multiple targets. You take the swabs and you preserve them and refrigerate them and then have them tested. And this just has to keep going on and on and on until you see something come up. And it could be that nothing ever comes up, or that is called asymptomatic shedding. And you're trying to catch asymptomatic shedding occurring on different parts of the body. And you really just have to guess where it might occur, where it might not. For like myself, for someone who has no visible symptoms, it'll be very interesting. We'll see, the asymptomatic shedding testing is then analyzed under PCR with a particular gene to target and determine if there are any false negatives occurring. And so that's just long-term testing that'll be ongoing for at least 3 months. And as the data comes in, we'll be reporting more and more on that data. But that's ultimately the only thing that'll really be able to show us what's happening in my body in terms of this virus itself.
Well, I know a lot of people are very eager to see that data, and I definitely wish you the best, and I hope it succeeds. And the herpes treatment that you injected in yourself at BDYHAX wasn't the first example of a public demonstration of injecting experimental treatment. We alluded to this earlier, but in October of 2017, Ford Fisher of News2Share live-streamed Tristan Roberts, who has HIV, injecting himself with an experimental HIV gene therapy treatment developed by Ascendance Biomedical. Do you have any updates on that front in the 6 or so months since that happened?
Yes. What we saw initially with Tristan was a very strong immune response for his CD4 and CD8 levels, a stronger response in terms of immune health, or indicative of positive immune health, than he had ever before seen in the course of his experience with HIV. His viral loads did increase, which may or may not have been a result of the treatment. Either way, they were still at a level where he could manage them without having to go on to antiretroviral medications, of which he does not take antiretroviral medications. He prefers to manage his disease through lifestyle and health and other strategies, and those work for him. The phase 2 is still being prepared. We are currently in the process of ensuring that the initial safety dose, which Tristan administered in October, that the core sequence can be translated 100% in terms of its identity to the 250 to 500 times stronger dose that is being prepared for mass production and distribution for emergency use in Venezuela upon confirmation of safety and expression. And we are moving forward as well with preclinical work to ensure that everything that we believe will result in terms of the long-term expression of the antibody at peak concentrations in the body will actually occur in the long term, 6 months or more.
You mentioned that there are plans to widen the distribution of this experimental HIV treatment to areas like Venezuela once it has been deemed safe. I imagine, or at least I hope, that some sort of peer review will be a part of that process. But have you determined what exactly is that you're looking for that would indicate this experimental treatment is safe?
Well, yes. N6 itself is a naturally occurring antibody within the body. The only issue is that it's very rarely ever at the peak concentrations necessary to actually give an individual the ability to fight off HIV all on their own. So the safety issues are very minimal when you're just working with something that is already a naturally occurring element in the body. However, it's a gene therapy, so you never know. Preclinical work is important to confirm not just that the N6 is expressing itself at the therapeutic levels in the body in perpetuity that we want it to, but also to ensure that there are no other negative elements that could be an issue. However, obviously for people in a country like Venezuela, this has been covered extensively by Guardian and by news outlets around the world since 2016. The people of Venezuela who have AIDS and the people who have HIV are literally just dying in hospital beds right now. That's it. The doctors have nothing, no medication to treat them. The doctors there barely even have the medication to treat things like the flu, let alone HIV or AIDS. So their only recourse is to lay there and die. And that is what is happening right now. So we will pursue the minimal effective dose of information required to ensure that what we are providing is safe, with the full knowledge that these individuals right now, in reality, are going to die because there are no options. We are not pursuing Venezuela to test a therapy. We are pursuing Venezuela because nobody else is. And if we have something that could save people's lives, and we are about as certain as humanly possible that there won't be the kind of adverse effects that would increase their currently terminal prognosis, then we are going to give this stuff away for free to anyone in that country who wants it. That is the only ethical decision. That is the only decision that could possibly be made. Anything else is an unethical, immoral, and just downright shameful usurpation of one's own responsibility to our fellow man and woman.
Shortly after Tristan Roberts injected Ascendance Biomedical's experimental HIV treatment during a Facebook live stream, the FDA released a statement that seems like it could be in direct response to that event, but it also could perhaps be referring to the work of Josiah Zahner at the Odin. I'll read a part of the FDA statement here. They said, FDA is aware that gene therapy products intended for self-administration and do-it-yourself kits to produce gene therapies for self-administration are being made available to the public. The sale of these products is against the law. FDA is concerned about the safety risks involved. Consumers are cautioned to make sure that any gene therapy they are considering has either been approved by FDA or is being studied under appropriate regulatory oversight. I'll post the link to the full statement in the show notes at futuregrind.org. But do you feel that this FDA statement was directed at your work, and how do you take that?
No, not at all. I mean, we are not selling compounds, and we have no intention of selling compounds that are being marketed for any sort of condition or with any sort of disclaimer that this can cure a disease or vaccinate against the disease. We're trying our best to get the data that shows that that's the case, but we can't make those claims outside of conducting a full-scale clinical trial. And we really wouldn't want to anyway, because they're never going to be conclusive for every single person and their individual experience of HIV or whatever other disease they may have that we're targeting. We just want people to have the opportunity to have access to research compounds that they can use for self-guided and self-directed research. And that's what access is about. And that's not illegal. That's been done for decades online with various other research compounds. And I would argue, and it won't be that hard for anybody out there to find proof of this, roughly 90% of the compounds out there that are currently being marketed as research compounds and that people are actively using for life extension purposes or purposes of, disease mitigation, these companies are not showing any sort of quality control documentation in terms of endotoxin levels, purity, or anything that any consumer, any individual would want to know before actually putting this stuff in their bodies. And so for us, we are committed to providing that transparency just as much as we are committed to providing that compound itself. And we do believe very strongly that the FDA should enforce purity and quality control standards, not just for the research compound industry and companies that are providing research compounds, but also for the supplement industry, which many people do not know is largely unregulated when it comes to GMP or good manufacturing practices in terms of the supplements that are produced. There are no disqualifying levels of arsenic or lead or other heavy metals in common over-the-counter vitamins that you could find anywhere at Walmart. And that is a problem. That is a major problem. And that should be addressed. And the FDA should move in that area and enforce a standard. There's no reason why they shouldn't. There's no reason why there can't be and shouldn't be quality control standards And those would, in fact, bolster the economy, because once you have quality control standards, you have to, as a manufacturer, work with laboratories to independently verify the quality control for the products or compounds or supplements that you are providing. And that creates more jobs in the sciences. And we need those. We deserve those. We're not concerned about the FDA. What we're concerned about is the FDA not moving in on the areas where it should have moved in decades ago, because it's such an easy win for them and such an easy win for Americans and the health of everyone.
You've received quite a bit of criticism over the past week. Some have said that your language could provide false hope to people dealing with these conditions. Some have said that your experimental treatments are irresponsibly dangerous and haven't been adequately tested. And some have claimed a lack of transparency and communication about what exactly is happening. And I think that you've addressed some of these concerns during our conversation today, but how do you respond to these criticisms? Is there anything that you took from this experience that might change how you do things in the future?
Yeah, I now know what it's like firsthand to have other people feel as though you shouldn't do what you desire with your body. I think it's certainly an experience that every woman in this nation and many nations around the world can relate to. And it's certainly one that individuals who are pursuing novel interventions in human augmentation and self-directed evolution will also encounter in the future. I think we've definitely covered a lot of the transparency. I think we've definitely cleared the base on that. In terms of that this could be something very dangerous, well, that's my danger. That's my risk. This is my body. And I have every right to assert my body in a way that does not harm other people. This was a completely 100% consensual act done between myself and myself. The world gets to see that, as they do with any research that's conducted in this way, and they deserve to see it. The question of false hope, well, that's just another way of saying the experiment isn't complete yet, and we don't know if it worked or not. And of course we don't, because that's why we do experiments.
I know that Ascendance Biomedical is in flux right now and is experiencing a period of transition, with some talent having recently departed the organization and a few question marks still hanging. What can we expect from Ascendance going forward?
Well, you can expect to see a very strong doubling down on 4 particular therapies that we will be expediting and focusing all of our work in over the coming months. You can expect to see us developing and producing the first large quantities of the gene therapy treatment for follistatin, which is human augmentation, effective for muscle mass, fat loss, and sarcopenia mitigation and bone mineral density loss in the aged, particularly for menopausal women. That will be the first application of our transfection platform that will be open access to researchers and entities around the world to be able to work with us in ways that we can catalyze that technology to actually serve even further disease states and disease conditions. You'll also see us continue forward with the N6 swiftly and effectively, hammer out all of the little minor issues that have kept us stalled for a little bit in terms of production, and move forward as quickly as humanly possible into Venezuela. Mid-March is the latest that we will be there. You can also expect to hear more about our work in PD-1 knockout for lung cancer, which we are currently in the beginning stages of initiating a clinical trial to support individuals who have lung cancer and who would like to receive that treatment offshore clinically. And you will also expect to see, of course, the data ongoing from my experimentation and work with the herpes experimental gene therapy-based cure and vaccine. And from that data, we will learn more about which specific production strategy we should optimize. And then we will move forward with producing the first 10,000 or so doses of that as well. All of this should be in place, and all of those things that I described should have occurred certainly by May 1st. I mean, that's just without a doubt. So in terms of a lot of our other work, we are just kind of going to scale back for a moment and focus really clearly on ensuring that these technologies that I just described can be optimized to the greatest degree possible, and that we can move forward on a very firm foundation as we transition back to some of our more comprehensive work with the microbiome, with aging in general and the Yamanaka factors, with cancer in general as well, and with other disease conditions.
So how big is the Ascendance team at this point, and who are you working with to make these projects a reality?
Well, we have our production partners, we have our researchers, some of whom prefer to remain unknown, and then we have a vast clinical network in our work in the fertility space with major clinics around the world for the delivery of that treatment. Yes, and obviously we have folks on our web development team and our investment team and everything of that nature, but we're less than 20 people. We always will be.
And speaking of the website, I know that you had a very interesting voting platform there at ascendance.io, in which you invited the public to help Ascendance choose your next project to tackle. Examples include hormone treatments, a quote, cocaine vaccine, and more. What was the idea behind the voting platform?
Well, it was just that's just kind of a fun thing. A lot of those are kind of just novelty projects that we thought the public might find interesting, and we just wanted to see what people were most interested in. And that would guide some of our next research, our next level research. And obviously what you saw there is that people are very interested in working with the hormones, particularly for transitioning from one gender to the next, or maybe just even for mediation of particular issues related to hormonal decline in age. And you can expect as well a very quick revamp on the website as well, most likely by the end of this month, whereby individuals will be able to see the methods and materials, the quality control documentation, and much more information regarding each of our therapies currently in development and the platforms behind them, so that individuals can have an opportunity not just to work with the platforms and the technology themselves, but to invest through inbound investing as well, and help to expedite the things that they want to see brought to reality the quickest.
We are nearing the end of our time together today, and there are a number of things that I would have loved to ask you that I haven't gotten the chance to, such as learning more about the idea of using blockchain technology and cryptocurrencies in your work with Ascendance. We'll have to get into that another time, but I guess one of the last questions I have is, what, if any, steps are you taking in your daily life to combat or slow the effects of aging? Are there any supplements, workouts, dietary strategies, or lifestyle changes that you've made?
Yes, absolutely. I recommend that individuals begin to learn how to fast in a way that does not stimulate loss of muscle. I recommend Tim Ferriss's section in Tools of Titans in his interview with Dr. Dominic D'Agostino, whose research into the effects of fasting with bodybuilders and sustained muscular growth is landmark and continues to be landmark. So I recommend experimenting with ketosis and ketogenic dieting. Though I don't recommend it as a 100% always do it kind of thing. I recommend metformin. I recommend rapamycin in very moderate intermittent doses. I recommend dasatinib and quercetin at just 5 days, following the same protocols that were used in the Mayo Clinic research with the mice, which has been confirmed by Dr. James Kirkland as effective in humans as well. He will not advocate for you to follow it. I will advocate for you to follow it. I have found it to be very effective. And also, we have affiliation with an independent pharmaceutical company that manufactures some of the greatest off-label treatments for the reduction of beta amyloid plaques from companies that unfortunately went under before they were able to bring those fully to market, not for reasons of science, but for reasons of bad business decisions. There are other things as well that people should communicate with me about, talk to me about. Obviously there are some things that, are best said publicly and other things that, best said in terms of personal consultation so that nobody gets hurt.
I appreciate your insight there. But I will remind the audience to make sure to thoroughly do your own research and consult your doctor before moving forward with anything that Aaron has just mentioned. It's always good to consult with the professionals and to get a second opinion. But you mentioned that people could reach out to you for more information. If someone would like to do that or would just like to follow along with your work, how would you suggest that they keep in touch?
Well, I'm on Facebook. There's only 2 Aaron Traywicks online right now that spell their name like mine, AARON TRAYWYCK. And, I'm the one with black hair. probably the prettier one, but I mean, I don't know. I've never met the guy. He might be an all right guy. He probably is. But so Facebook is easiest, honestly. Just friend me on Facebook or send me a message on my public profile. You can always go to the website and fill out any one of our forms, and we'll do our best to get in touch through there. But if you want a quick answer, if you want a quick response, just get at me on Facebook. That's where I like to do most all of my business, or our business. It's just so much easier and faster than most other things. It's got its flaws, obviously, but it's fast and it's a pretty effective platform.
Well, thank you again for taking the time to speak with me. There have been a lot of things said in the media recently about you and your work, and I know that more are scheduled to come out in the coming weeks. So this is a topic of conversation that will continue, and I thought it was important to hear directly from you. My hope is that this will be a resource for others to be able to find, to learn more, and make up their own minds. So, Aaron, stay safe and best of luck.
Absolutely. I couldn't agree more, and I was glad to do it. And thank you so very much for having me here.
Hey everyone, Ryan O'Shea again, and thanks for listening to my interview with Aaron Traywick. One more reminder that self-administering any type of unregulated gene therapy or other treatment is risky. Anything that Aaron mentioned during this interview should be viewed as experimental research compounds not intended for human use. I encourage you to consult your doctor with any questions. Remember to view the show notes and more at futuregrind.org. You can also like, comment, and share to spread the word. Till next time, this is Future Grind.